Microbial physiology. So what is physiology? Physiology, it is the study of the vital life processes of an organism or how an organism function. So when we and microbial, from its root word microbes, it means those are the organisms that cannot be seen by the naked eye but with the use of a microscope. So when we say microbial physiology, it is the study of the life processes of the microorganism. So why do we need to know, or why do we need to study the microbial physiology? Because scientists can learn a great deal about cells, including human cells, by studying the nutritional needs of bacteria, their metabolic pathways, and why they live, grow, multiply, or die under certain conditions. So if bacteria, because microorganisms are almost everywhere, they are everywhere, and they reproduce rapidly, so that's why they are prone to researches than human cells. Microbial nutritional requirements. To build necessary cellular materials, every organism requires a source of energy, a source of carbon, and additional nutrients. Those materials that organisms are unable to synthesize but are required for the building of macromolecules and sustaining life are termed essential nutrients. Essential nutrients, that is, essential amino acids and essential fatty acids, must be continually supplied to an organism for it to survive. Essential nutrients vary from species to species. Categorizing microorganisms according to their energy and carbon sources. Various terms are used to indicate an organism's energy source and carbon source. Terms relating to an organism's energy source. The terms phototroph and chemotroph pertain to what an organism uses as an energy source. Phototrophs use light as an energy source. The process by which organisms convert light energy into chemical energy is called photosynthesis. Chemotrophs use either inorganic or organic chemicals as an energy source. Chemotrophs can be subdivided into two categories: chemolithotrophs and chemoorganotrophs. Chemolithotrophs, or simply lithotrophs, are organisms that use inorganic chemicals as an energy source. Chemoorganotrophs, or simply organotrophs, are organisms that use organic chemicals as an energy source. Terms related to an organism's carbon source. The term autotroph and heterotroph pertains to what an organism uses as a carbon source. Autotrophs use carbon dioxide as their sole source of carbon. Photosynthetic organisms such as plants, algae, and cyanobacteria are examples of autotrophs. Heterotrophs are organisms that use organic compounds other than carbon dioxide as their carbon source. Humans, animals, fungi, protozoa are examples of heterotrophs. The terms relating to energy source can be combined with the terms relating to carbon source, thus yielding terms that indicate both an organism's energy source and carbon source. For example, photoautotrophs are organisms that use light as an energy source and carbon dioxide as a carbon source. Photoheterotrophs, such as purple non-sulfur and green non-sulfur bacteria, use light as an energy source and organic compounds other than carbon dioxide as a carbon source. Chemoautotrophs use chemicals as an energy source and carbon dioxide as a carbon source. Chemoheterotrophs use chemicals as an energy source and organic compounds other than carbon dioxide as an carbon source. All animals, all protozoa, all fungi, and most bacteria are chemoheterotrophs. All medically important bacteria are also chemoheterotrophs. Metabolic enzymes. The term metabolism refers to all of the chemical reactions that occur within any cell. These chemical reactions are referred to as metabolic reactions. The metabolic processes that occur in microbes are similar to those that occur in cells of the human body. Metabolic reactions are enhanced and regulated by enzymes known as metabolic enzymes. A cell can perform a certain metabolic reaction only if it possesses the appropriate metabolic enzyme, and it can possess that enzyme only if the genome of the cell contains the gene that code for the production of the enzyme. Biologic catalyst. Enzymes are known as biologic catalysts. Enzymes are proteins that catalyze or speed up or accelerate the rate of biochemical reactions. In some cases, the reaction will not occur at all in the absence of the enzymes. Enzymes are very specific. A particular enzyme can catalyze only one particular chemical reaction. In most cases, a particular enzyme can exert its effect or act on only one particular substance known as the substrate for the enzyme. Enzymes produced within a cell that remain within the cell to catalyze reactions within the cell are called endoenzymes. The digestive enzymes within phagocytes are a good example of endoenzymes. They are used to digest materials that the phagocytes have ingested. Enzymes produced within a cell that are then released from the cell to catalyze extracellular reactions are called exoenzymes. But some proteins, called apoenzymes, cannot, on their own, catalyze a chemical reaction. An apoenzyme must link up with a cofactor to catalyze a chemical reaction. Cofactors are either mineral ions, example, magnesium, calcium, or iron cations, or coenzymes. Coenzymes are small, organic, vitamin-type molecules. Factors that affect the efficiency of enzymes. Enzyme efficiency is influenced by various factors including pH, temperature, and the concentration of the substrate. An enzyme will function at peak efficiency over a particular pH range. If the pH is too high or too low, the enzyme will not function at peak efficiency. Likewise, an enzyme will function at peak efficiency over a particular temperature range. If the temperature is too high or too low, the enzyme will not function at peak efficiency, and the reaction that the enzyme catalyzes will not proceed at its maximum rate. Substrate concentration is another factor that influences the efficiency of an enzyme. If the substrate concentration is too high or too low, this enzyme will not function at peak efficiency, and the enzyme catalysis will not proceed at its maximum rate. Going back to metabolism, so most metabolic reactions fall into two categories: catabolism and anabolism. So first, let us know what is a metabolite. Metabolite is any molecule, either a nutrient, an intermediary product, or an end product in a metabolic reaction. So, catabolic reactions involve the breaking of chemical bonds and the release of energy, while anabolic reactions involve the formation of bonds which requires energy. The key point about catabolic reactions is that they release energy. Catabolic reactions are often referred to as degradative reactions because they degrade larger molecules down into smaller molecules. Biochemical pathways. Biochemical pathways is a series of linked biochemical reactions that occur in a stepwise manner, leading from a starting material to an end product. Two common processes are the biochemical pathways known as aerobic respiration and the fermentation reaction. Aerobic respiration of glucose. The complete catabolism of glucose by the process known as aerobic respiration occurs in three phases, each of which is a biochemical pathway: A. Glycolysis, the Krebs cycle, and the electron transport chain. Although glycolysis is an anaerobic process, while the other two phases require oxygen, hence it was named aerobic respiration. So glycolysis, also known as the glycolytic pathway, the MD-Meyer-Hof pathway, and the MD-Meyer-Hof Parnas pathway, is a 9-step biochemical pathway involving 9 separate biochemical reactions, each of which requires a specific enzyme. In glycolysis, a 6-carbon molecule of glucose is ultimately broken down into 3 carbon molecules of pyruvic acid, also called pyruvate. Glycolysis can take place either in the presence of oxygen or in the absence of oxygen. Oxygen does not participate in this phase of aerobic respiration. Glycolysis produces very little energy, a net yield of only 2 molecules of ATP. Glycolysis takes place in the cytoplasm of both prokaryotic and eukaryotic cell. Krebs cycle. The pyruvic acid molecule produced during glycolysis are converted into acetyl coenzyme molecule which enter the Krebs cycle. The Krebs cycle is a biochemical pathway consisting of 8 separate reactions, each of which is controlled by a different enzyme. In the first step of Krebs cycle, acetyl coenzyme A combines with the oxaloacetate to produce citric acid. Hence, the other name for the Krebs cycle, the citric acid cycle. It is referred to as a cycle because at the end of the 8th reaction, the biochemical pathway ends up back to its starting point, the oxaloacetate. Only 2 ATP molecules are produced during the Krebs cycle. So, the electron transport chain. As previously mentioned, certain of the products produced during the Krebs cycle enter the electron transport chain. The electron transport chain consists of a series of oxidation-reduction reaction whereby energy released as electrons are transferred from one compound to another. Those compounds include flavoproteins, kinones, non-heme iron proteins, and cytochromes. Oxygen is at the end of the chain. It is referred to as the final or terminal electron. So, the fermentation of glucose. The first thing to note about fermentation reactions is that they do not involve oxygen. Therefore, fermentation usually takes place in anaerobic environments. The first step in the fermentation of glucose is glycolysis, which occurs exactly as previously described. Remember that glycolysis does not involve oxygen and very little energy is produced by glycolysis. The next step in fermentation reaction is the conversion of pyruvic acid into an end product. whereas oxidation reactions involve the loss of an electron, whereas reduction reactions involve the gain of an electron. On the other hand, anabolism refers to all the anabolic reactions that occur in the cell. Anabolic reactions require energy because chemical bonds are being formed. Most of the energy required for anabolic reactions is provided by the catabolic reactions that are occurring simultaneously in the cell. So example of anabolic reactions include creating a disaccharide from two monosaccharide by dehydration synthesis, the biosynthesis of polypeptides by linking amino acid molecules together, and the biosynthesis of nucleic acid molecules by linking nucleotides together. Biosynthesis of organic compounds. The biosynthesis of organic compounds requires energy and may occur through either photosynthesis or chemosynthesis. So photosynthesis, light energy is converted to chemical energy in the form of chemical bonds. So the goal of photosynthetic process is to trap the radiant energy of light and convert it into chemical bond, which can be converted into more ATP molecules at a later time through aerobic respiration. So the chemosynthesis, this process involves a chemical source of energy and raw materials for synthesis of the metabolites and macromolecules required for growth and functions of the organisms. Bacterial genetics. So genetics, it is the study of heredity, example, DNA, genes, the genetic code, chromosomes, DNA replication, transcription, and translation, which we all know that already. An organism's genotype or genome is its complete collection of genes, whereas an organism's phenotype is all the physical traits, attributes, or characteristics of the organism. So, phenotype is the manifestation of genotype. The genes direct all functions of the cell, providing it with its own particular traits and individuality. So, it is said that the gene coding for the particular gene product is said to have been expressed. All the genes on the chromosomes are not being expressed at any time because it will be a waste of energy. So the genes that are expressed at all the times is called constitutive genes, and those that are expressed only when the gene products are needed are called inducible genes. Mutation. So mutations, it is the alteration of the gene, but the gene doesn't die. Instead, it just alter, and it is transmitted to the daughter cells of each succeeding generation. A change in the characteristic of cell caused by a change in the DNA molecule that is transmissible to the offspring is called mutation. So there are three categories of mutation: the beneficial mutations, harmful and sometimes lethal mutations, and silent mutations. So beneficial mutations, as the name implies, are of the benefit to the organism. So example, the gene mutates in order for it to survive. So perhaps the mutation enables the organism to be resistant to a particular antibiotic. An example of an harmful mutation would be a mutation that leads to the production of a non-functional enzyme. So a non-functional enzyme is unable to catalyze the chemical reaction that it would normally catalyze if it were functional. So the silent mutation, that is, they have no effect on the cell. Physical or chemical agents that cause an increased mutation rate are called mutagens. The organism containing the mutation is called a mutant. There are at least 4 additional ways in which the genetic composition of bacteria can be changed. The lysogenic conversion, transduction, transformation, and conjugation. So, lysogenic conversion. In lysogenic conversion, the bacterial cell will exhibit new properties, a phenomenon known as lysogenic conversion. So in other words, the bacterial cell has been converted as a result of lysogeny and is now able to produce one or more gene products that it was previously unable to produce. So a lysogenic bacterium is capable of producing one or more new gene products as a result of infection by a temperate bacteriophage. Transduction. So in transduction, it also involves bacteriophages. Transduction means to carry across. Some bacterial genetic material may be carried across from one bacterial cell to another by a bacterial virus. Only small segments of DNA are transferred from cell to cell by transduction compared with the amount that can be transferred by transformation and conjugation. Transformation. So in transformation, a bacterial cell becomes genetically transformed following uptake of DNA fragments or naked DNA from the environment. Conjugation. The process of conjugation was discovered by Joshua Lederberg and Edward Tatum in 1946 while experimenting with E. coli. So in conjugation, genetic material usually in the form of a plasmid is transferred through a conjugative pore from a donor cell to a recipient cell. A plasmid that contains multiple genes for antibiotic resistance is called a resistance factor or R factor. Transduction, transformation, and conjugation are excellent tools for mapping bacterial chromosomes and for studying bacterial and viral genetics.